I got into science because I liked biology.
Uh-huh.
I liked watching the nature shows. And in high school, when we did the DNA replication with the Okazaki fragments, I thought that was cool. I was like, oh, that's how you do that. So I think that's when I realized I liked the more molecular biology rather than the organismal biology. So how did I get into biology? Oh, that was kind of how I got into biology. From this biology class in high school, I knew I liked the more molecular basis of research because it seemed like you got real answers rather than. So I picked a college program that had research available for undergraduates in the biochemistry program. And so that's how I picked a school, and I also knew I wanted to go to graduate school because I had another biology teacher, Mrs. Beatty, who had a chart about all the different careers you could do with biology. And then she had a graph like, this is how many students get an undergraduate degree in biology. This is how many students get a master's degree. This is how many students get a Ph.D. I was like, oh, okay, I'll get a Ph.D. I was still doing it. So the yeast biology, I took a class called molecular genetics. So when I started doing research, I wanted to, make an impact on human medicine and, make my contribution to humanity. So I was in a psychology lab studying mechanisms of neurodegeneration in addiction, particularly alcoholism. And it was an older style lab, so very biochemistry, a lot of enzymology. And then I took this molecular biology class and met Dr. Lenore Nygborn. She was one of the professors. And I really liked her, and I liked, her enthusiasm and energy. And then I kind of decided that I wanted to try something different. Genetics with Lenore. And decided that I wanted to try something different. So that summer, I went to Cornell Medical School for a research program, and this was the first time I had a project, and I worked all day in the lab, and that seemed like, oh, you could finally see an experiment from the beginning to the end. So when I came back, I switched to Lenore's lab, where she studied yeast meiosis and was doing a genetic screen looking for other genes that interacted with this kinase that she was very interested in. And unfortunately, she didn't get tenure. So I spent the summer in her lab after graduating, finishing up some projects. Went to my first yeast conference in Maryland. That was overwhelming. There was 1,000 people. And I presented my poster on the project I was doing. And then I went to graduate school. So the way I picked the graduate school, I was talking to my first research advisor, and I was like, oh, I'd like to go to graduate school. Where do you think I should apply? And I was thinking, another state school, It's like, well, why don't you try Harvard and Yale? I was like, well, I can't get in there. He's like, of course you can. It's like, oh. I couldn't get in there as an undergraduate, but doing research is different. You have a different skill set. It's more driven by curiosity and having to deal with the frustration of doing the research and, just that little peak of hope that something will come out and you'll figure something out. It's not like just getting a good grade on an exam. You take the exam, it's done, and then you can forget everything, and then it's okay. But in research, you have to keep building upon what you know. So when I interviewed for schools, I looked for schools that had genetics labs. The students seemed happy because I didn't want to go to a place where everybody was so stressed out. That was a small program. So the program I was in was 12 students in my class.
In Yale?
At Yale, yes. Like at Berkeley, I know they have 50 students in the class, and I was like, oh no, that's too big. I don't want to go to a program that big. So the kind of research that I'm doing now is studying genetic variation. So individual people are genetically different, and if you expose them to the same environment, they will respond differently to that environment. And what makes us different from you and me is our genes and how we use our genes. So we're looking at genetic differences between people. So one way you could address that is you take a bunch of different people, you expose them to different environments, and you measure all that stuff. But, but we are very complicated organisms. We have a lot of genes, we have a lot of regulatory regions. So how do you figure out what's important? Well, what we do is we look at a much simpler organism that has fewer genes and only one cell, because in individuals, your kidney cells act differently than your liver cells based on the kinds of genes they have, and maybe your liver and somebody else's liver acts the same, but your kidneys react differently to a different environment. So how do you tease out what is coming from your kidneys that are different, but your liver acts the same? So one way to do that is just to avoid the whole problem and work in a single-cell organism. So I work in Saccharomyces cerevisiae, which is the same yeast that makes beer and bread. So it's a very well-domesticated yeast, probably the first organism that has ever been domesticated.
Yeah.
And yeast do many of the things that we do. They have to make proteins, they have to duplicate their DNA, they have to divide, they have to make offspring. So we can use this as a model to look at what's important for reacting to different environments. And because we have the entire genome sequenced of lots of different cerevisiae, we can pinpoint exactly what we're looking at, and also we can manipulate those genes a lot easier. Like, you probably don't want to have your Cre recombinase introduced to you to knock out, your PKC genes. Probably would not be happy with that. But a yeast, it doesn't care. I mean, it's going to be a little cranky probably. Now, unfortunately, there are certain skills now so the field has changed from when I started. When I started, I worked on a single gene, and I knew everything about that one gene. But now we realize that while one gene can be very important, you need to look at the organism as a whole, because everything interacts with each other. If I studied one thing that was involved in the nucleus, I completely ignored everything that goes on in the ER. But now we know that if you have some stresses in the nucleus, that can affect the ER and the ER. Everything is connected. So programming would probably be greatly beneficial as statistics. That is now something I wish I had taken statistics, but more rigorous. Because now what a lot of the data is a spreadsheet. And how you determine what is important is by a threshold. And how do you determine where that threshold is? Well, you have to look at statistics. And so I have some very useful collaborators that help me figure this out. So the experiment today was to look for genes that, if they are no longer present in the cell, does the response to this drug change? Because we found targets, transcriptional regulator targets by year 1, and we have a collection where all genes are knocked out. So you have, probably it's only like 4,000 different tubes, and in each tube is a different yeast strain that has a different gene removed. So I went and collected those strains, those candidates, and put in the different alleles of year 1. And I grew them up overnight, and then I spotted them down onto plates with drugs. So tomorrow I'll come in and take a picture to see how well they grew.
That's Saturday.
Yes, it's Saturday. But it would be much easier for me to come in on Saturday and do an hour worth of work than, say, on Thursday, it's Friday. If I do this experiment now, then I'll have to come in on Saturday, so I won't do it. So then I wouldn't have done anything today. So the nice thing about research is that you decide what's interesting, and you work on your own time scale. I also tend to work really late, so I came in probably at 10:30 today. It's Friday, so I'll probably leave a little early, but usually I'll leave around 8, 10, 12. I realize leaving at 12 is a bad idea because I start to get really tired.
When do you eat dinner?
I don't eat dinner. I eat a really big lunch with all my lab friends. That's my main meal. So in the afternoon, I'll have coffee. I have snacks at my desk. I have some nuts, some lovely chocolate. Well, we did have an incident in the lab once where, so we want to make sure everything is sterile when we're doing things, and so we sterilize them by dipping them in alcohol. But you don't want to sit there and wait until the alcohol evaporates, so you set it on fire. So that's called flaming. So, we'll flame our pipettes, the tops of bottles, to make sure that they get sterile and stay sterile. So there was a postdoc in the lab who was transferring small amounts, and so he had remember that frog that I showed you with the pins yesterday? Oh, sorry. So this is a metal device that has 48 pins on it, and you put it into a dish that has yeast on it, and the yeast stick to the pins, and then you can easily transfer 48 different little tubes, like yeast strains at once, and put it onto like a Petri plate. And you just, can keep going back into that original dish that has the 48 wells. Well, he was flaming it. He had a dish of alcohol, and he accidentally put the pinner that was still on fire into the alcohol and set the whole alcohol bath on fire. And so I was not in the room. I came back in, and I see 3 people standing in front of his bench, and I walk over. I'm like, what's going on? They're like, well, the alcohol's on fire, and we don't really know how to put it out. Like, well, it's a fire. Don't you use a fire extinguisher? I'm like, oh, yeah. So one of the other postdocs was like, well, when I was in graduate student, I took a special course on how to use a fire extinguisher. We're like, okay, then you do it. It wasn't a good idea, because what happened was the fire extinguisher shoots that puff of whatever it is, and then it pushed out all the ethanol out of the bath onto the bench and set everything on fire. And then styrofoam is melting, and I'm just like, oh crap. So we had a lounge that had a blanket. So I ran in there and I got the blanket and I put it on top of the bench and smothered it. So don't use a fire extinguisher to put out an ethanol bath. You can, get some heavy, like the other time I set something else on fire, I used an autoclave glove, which is a very heavy mitt, for getting hot stuff out of the autoclave, and put that on. It just smothered it. So that was, I guess he was a little embarrassed. He set the lab on fire. And then we called the fire department because we're like, well, there's all this smoke in the lab, but the fire alarm didn't go off. And so we're explaining to the fire people, like, oh, well, this has happened. They're like, well, pull the fire alarm. We're not pulling the fire alarm. There's no fire. And everybody's going to get pissed at us because then they're going to have to leave their lab. Because that's really annoying when you're in the middle of an experiment and the fire alarm goes off. Because then I was in another building. It was a brand new building. And they hadn't finished it when we moved in, so there was still construction. And the fire alarm would go off about once a week. Usually Tuesdays between 2 and 4, because that's when we had lab meeting. Sometimes it would go off multiple times, but they always claimed it's not a false alarm. The alarm went off because, they decided to put the autoclave in a room with a fire alarm right in front of it. And when you open the autoclave, steam comes out, and what is that going to do? It's going to set off the fire alarm. So that would also set it off. They were doing construction, and the dust would set it off. And so it got to the point where people were getting too annoyed because they're like, there's no fire. So they wouldn't leave the lab anymore. And then the fire department got pissed because they're like, the fire alarm is going off.